Okay, so welcome to this next video on the growth factor receptor and MAPK ERK pathway. Okay, right. So far what we got to was that in our cell that has been stimulated by the growth factor receptor, we now have an activated mitogen-activated protein kinase, or MAP kinase, which is a MAP kinase with a phosphate group up there. So this is the activated MAP kinase, or mitogen-activated protein kinase, or microtubule-associated protein kinase, or it's sometimes also called ERK, for extracellular signal-regulated kinase. So this is our activated MAPK, MAPK slash ERK protein. And we've seen that one of its actions is to phosphorylate the transcription factor MYC, basically. Now what we're going to do is look at its another one of its actions. And basically, for this, it needs to translocate into the nucleus of the cell. So let's show it translocating into the nucleus. So let's draw the nucleus out here. Okay, so this will represent our nucleus of our cell, and basically what it's going to go and do is the activated MAPK, MAP kinase, is going to go into the nucleus and it's going to phosphorylate another transcription factor. And this other transcription factor is a transcription factor by the name of ELK1, so ELK1. So what's going to happen is you're going to take the ELK1, which is unphosphorylated. MAP kinase is going to phosphorylate a serine or a threonine residue in that ELK1 protein, and you're going to get a phosphorylated ELK1. Now, the phosphorylated ELK1 is the active ELK1. So ELK1 is a transcription factor, so when it's activated, what it's going to go and do is it's going to go and bind to a certain portion of the DNA, and it's going to bind basically at promoter regions of genes. So ELK1 is going to go and bind to a promoter region here, and it's going to activate the transcription of certain genes by increasing the affinity of the DNA for RNA polymerases, basically. Okay, so here is a promoter region in green, and the gene is in pink. So ELK1 is going to go and bind to the promoter region here. And one of the genes that specifically it's going to increase the expression of is a gene for C-phos. So this is the C-phos gene. Okay, so what happens is that ELK1, with its new phosphate group attached to it, is going to bind to the promoter region of the C-phos gene and is going to increase the transcription of this gene. Okay, so that means you produce more mRNA, and if you produce more mRNA, you're going to get more translation of that mRNA into the active protein, so you end up with more C-phos protein. Okay, now what is C-phos? C-phos is basically another transcription factor, but it's not a transcription factor on its own. It has to dimerize with another protein, which is called C-jun, and together what they make is a jun-phos complex. So basically, if you activate ELK1, it's going to result in the production of C-phos, and C-phos is going to dimerize with C-jun to make a phos-jun dimer, and phos-jun dimer is a very, very powerful transcription factor, basically. Okay, so equally, it's very similar. It's going to have a very similar effect to MYC, C-MYC, basically. So now what we're going to do is reconverge those two pathways. So we saw that the activated MAP kinase catalyzes this reaction with, this phosphorylation of the C-MYC protein, which was also a transcription factor. So here is our C-MYC protein being phosphorylated, basically. So we've now got C-MYC with a phosphate group on it. Okay, and now these two transcription factors, C-MYC and phos-jun dimers, those are going to pretty much do the same thing in the cell. So by these two different pathways, you overall get transcription factors which are going to do pretty much the same thing. Okay, so let's draw other genes here. Okay, so these are genes which are going to be turned on by our phos-jun or dimers or our C-MYC protein. Okay, so we have our DNA here, and we'll have a promoter region here, and a gene here. So these phosphogen dimers, or this c-MYC transcription factor, are going to bind to the promoter regions of loads of different genes here, and it's going to increase or decrease potentially the transcription of the gene. Okay, so basically what happens is that when c-MYC and phosphogen dimers go up in the nucleus of the cell, you get an increase in transcription of loads of genes which will produce proteins that are necessary in order to replicate the DNA. Now that is the characteristic of G1 phase of the cell cycle. G1 phase, remember, of the cell cycle was about producing all the proteins that are necessary in order to replicate the DNA, i.e. preparing basically for S phase of the cell cycle. So, if we have activated the production of proteins associated with replication, we are now going to go into G1 phase. Okay, so these transcription factors are going to lead to the increase in expression of proteins needed for DNA replication. Proteins needed for DNA replication. Okay, so you're going to take the cell into G1 phase of the cell cycle, DNA replication. Now, again, just like in the case of the Wnt beta-catenin pathway, these transcription factors are also going to start increasing the transcription of proteins associated with actually beginning the S phase. So proteins associated with taking you from G1 into S phase, i.e. getting you past that G1S checkpoint, basically. So what we've seen so far is that these transcription factors, phosphogen dimers and c-MYC, are causing an increase in the transcription and therefore expression of loads of proteins associated with DNA replication. So you're getting all of the stuff ready to replicate your DNA. Now, to actually start the replication of the DNA, you need to create some other proteins. There are some proteins in this preparatory process that are kept low right until the end, and without them you can't actually begin the DNA synthesis phase. So in order to actually get those final proteins made, you need to activate a different set of transcription factors, not phosphogen or c-MYC, but basically phosphogen dimers or c-MYC are going to activate the proteins which activate those transcription factor. Okay, so let's see that pathway. Right, so another protein basically that phosphogen dimers and c-MYC is going to increase the expression of is the Cyclin D protein. And Cyclin D is what's going to get the cell ready to jump from the G1 phase to the S phase. So Cyclin D is a protein, and when it goes up in the cytoplasm of the cell, it binds to its enzyme, and the enzyme which it binds to is called cyclin-dependent kinase 4, which is often abbreviated to CDK4. So this is cyclin dependent kinase 4. Dependent kinase 4. Okay, and this enzyme is a kinase, so it's going to add phosphate groups onto things, and it's also often abbreviated to CDK4. So usually in the cytoplasm, when you haven't got any cyclin D, this cyclin-dependent kinase 4, the CDK4 enzyme, is inactive. However, when cyclin D binds to it, what happens is you form a structure known as a cyclin D CDK4 complex. So here's cyclin D bound to the CDK4. And this entire complex now is known as a cyclin D CDK4 complex. Cyclin D CDK4 complex. Okay? And this complex, its role in the cell is now to phosphorylate a protein known as the retinoblastoma protein. Okay, so now we need to talk about the retinoblastoma protein for a little while. So, as we've seen, these signals that are going to get the cell to move into the S phase from the G1 phase, they're producing cyclin D. Cyclin D is activating CDK4, so CDK4 activity will go up in response to these growth factors, basically. So, what we're now going to look at is what is usually stopping the cell from going into the S phase, basically, from starting DNA replication. Well, basically, it's a protein known as retinoblastoma protein, which, for a cartoon, I will draw like so. So here is the retinoblastoma protein. So this is retinoblastoma protein, because it was discovered to be mutated in retinoblastomas. Okay, so it's often abbreviated to just RB, rather than having to write the full name out, retinoblastoma protein, so RB. So RB's role in the cell is to bind to a very powerful transcription factor, which is made up of a dimer, basically. So this powerful transcription factor is a dimer of proteins. One of them is something known as an E2F transcription factor, and the other is known as a dimerization partner of E2F transcription factors. Excuse me. So you get, in the cytoplasm of the cell, there is this dimer of proteins, which is an E2F transcription factor dimerized with its dimerization partner protein. So this is the dimerization partner for E2F transcription factors, dimerization partner. Okay, and basically this dimer of E2F with its dimerization partner is an extremely powerful transcription factor which will go to the DNA and bind to promoter regions and increase the transcription of genes encoding for proteins, which will actually begin the replication of the DNA. Right, so usually when a cell is not dividing, the retinoblastoma protein has the E2F protein transcription factor with its dimerization partner firmly held to it. Okay, so inactivating it, basically stopping it from activating the transcription of these proteins. So if you want to go from the G1 phase to the S phase, what you need to do is inactivate this retinoblastoma protein so that the E2F transcription factor with its dimerization partner can be released and go off to the nucleus and activate transcription. And that's exactly what the cyclin-dependent kinase 4 with cyclin D bound to it does. So this cyclin D CDK4 complex phosphorylates this retinoblastoma protein here. And when it phosphorylates the retinoblastoma protein, what happens is the retinoblastoma protein changes conformation, like so. So I'll say imagine that it's moved these outward, and then it's shrunk a little bit as well because I haven't got enough space. But it changes conformation, and now basically it releases the E2F protein dimerized with its dimerization partner here. So here's its dimerization partner, here's the E2F transcription factor. Okay, so now this dimer is going to go into the nucleus, so here it goes into the nucleus, and it's going to activate the transcription of a bunch of genes by binding to promoter regions which have a certain binding site for this E2F dimerization partner complex, which is known as an E2F box. So certain promoter regions will have what is known as an E2F box in them, where this E2F DP complex can bind. So this is the E2F box. And basically, when it binds to the E2F box, it increases the transcription of the downstream gene over here. So I'll draw that in green. I've got them the wrong way around from the previous color coding, but never mind. So it's going to cause an increase in the transcription of the gene, and basically these are the final this gene is going to be transcribed and then translated, and it will give the final proteins that are essential for DNA replication to begin. So these are the final ones that you need. You've built most of them already, but there are some that are missing, and this is going to give the final ones, and then that is the signal for DNA replication to actually start. So in order to go just from the G1 phase, where you're just accumulating the necessary enzymes, the DNA polymerases, etc., to a phase where you're actually replicating the DNA, you need to produce cyclin D to activate the CDK4 enzyme, and then inactivate retinoblastoma protein and activate the E2F DP complex. And basically, these phosgen dimers and the c-Myc transcription factor, those are capable of increasing the transcription of cyclin D. So, overall, what happens? Growth factors binding to their growth factor receptors, lead to the production of active phosphogen dimers and c-MYC. These transcription factors increase the expression of the enzymes associated with DNA replication, i.e. they take the cell from interphase to the first growth phase. They also increase the transcription of Cyclin D, which gets you ready to move from G1 phase to S phase. So they basically kickstart the cell cycle. They replicate the DNA, and after that, you're ready to actually divide and produce another cell. So these are powerful stimuli to cells.